Within OncoGEO, users can utilize three views to view the distribution of data within the land. These can be found under Select View, Sample Distribution, Comparison Distribution, and Project Distribution. The default view for any land that's opened within Array Studio is the Sample Distribution view. In this view, all samples within the land are plotted in a stacked bar graph. These samples are grouped on the y-axis based on disease category and colored on the x-axis based off of sample type, such as normal or tumor samples. Users can quickly toggle to the other distribution views by going to Select View, Comparisons. Comparisons view shows all comparisons that have been curated by Omicsoft scientists within the land. These comparisons are grouped on the y-axis based on tissue category and colored on the x-axis based off of the type of comparison, such as disease versus normal, drug-resistant versus sensitive, or comparing different tissue or cell types. The third distribution view under Select View is Projects. The Project Distribution view will list all projects contained within the land, grouped based off of specific clinical focus. In all distribution views, selecting the bar within any of the graphs will populate Details view for the data selected. In this example, we have selected immunomodulators. This therapeutic area is related to all immune therapy related projects. In the Details view, we can find all related information in the metadata, including the type of therapy provided and additional metadata. Note that projects can belong to more than one therapeutic area, and this is controlled by the therapeutic area metadata column. Let's use the sample distribution view to find samples related to the anti-PD-1 therapy study. There are a number of ways to identify samples within this view. First, under the Sample tab, we can use information from the associated study. For example, the anti-PD-1 patients were treated for metastatic melanomas. Under the Metadata column, for disease category, we can find melanoma or skin cancer and select that. Under Land Sample Type, we can search for metastatic samples and right-click and select Check This Only. The view will instantly be updated to reflect the metastatic skin cancer samples. In addition to using the metadata to filter for samples of interest, we can use the metadata groupings to group our samples in this view. Under Grouping, all metadata will be available. Let's see how many projects were associated with metastatic skin cancer within OncoGEO. By using project name, we can group by the number of projects that were present within this land. Typing project will populate the metadata column, project name. Selecting this column, we see our chart is instantly updated to show that there are 5 projects that were with metastatic melanoma patients. These are grouped by their GSE accession number. One of the 5 projects, GSE 78220, corresponds to the anti-PD-1 related study. Clicking on the bar in this group will populate the Details view with all samples used in this study, as well as associated metadata. In addition to the details, we can use filter selection to filter our samples to just samples from this project. Another simple way to filter to samples from this project is to use the Project tab and use the string filter by clicking on the radio button and typing in the GSE accession number. Clicking OK will instantly update our filters to select for just this project. Project information is carefully curated by Omicsoft scientists and can be used to group samples within the study. For example, since this project compared anti-PD-1 responders to non-responders, let's again use the grouping to group our samples by response. Simply typing in the first few letters for response will filter our metadata. Let's use response to group our samples. Using this grouping, we can see that the data has been grouped into 3 sample groups for this project: progressive disease, or patients that are resistant to anti-PD-1 therapy, as well as samples from patients that are both partially or completely responding to anti-PD-1 therapy. Clicking on the bars corresponding to samples from these groupings, we can see that there were 13 samples from anti-PD-1 resistant, 10 from partially responding, and 5 from completely responding. Selecting outside of the graph will deselect and remove the details view. A useful view in the land is the survival view. Under Select View, Survival Data. In this view, a Kaplan-Meier survival plot will show survival of individuals with our designated groupings. It is clear from this plot that the responding groups live longer than the non-responding group. Under Task, we can customize this view. For example, we can change line properties and adjust the width so that the width of these lines is easier to view. We can also show summary information. This will show results from a statistical significance perspective, showing that these responding patients live longer than the non-responding patients.